Namaskar दोस्तों, welcome to channel Toxicology on Board. Today we'll discuss question number 39, DABT question. The topic is Comet Assay, OECD 489 guideline and genotoxicity. So let's directly dive into the question. The single cell gel electrophoresis assay, Comet assay, is used to detect what genotoxic endpoints. The question is Comet assay used to detect what genotoxic endpoints. Okay, let's see we have what are the options given in the question. Option A is single and double strand DNA breaks in cells and tissue. Option B is point mutations in bacterial and mammalian cells. Option C is sister chromatid exchanges in mammalian cells. Option D is aneuploidy in bone marrow cells. The right answer is option A, and an explanation and reference which is given by ABT is actually the question is I forgot to say that the question is from American Board of Toxicology only, okay, from their quiz. Okay, the right answer is single and double strand DNA breaks in the cell and tissue. So Comet assay used to identify this type of damage. Okay, the Comet assay measures isolated DNA fragments using a gel electrophoresis method, right? Smaller fragments migrate further in the gel and length can be measured. So if you see the smaller this is the bigger DNA, larger DNA. These are the smaller fragments which are migrating in the gel electrophoresis. By measuring the length of this migration, we can identify the amount of damage that particular chemical causes. So it says smaller fragments migrate further in the gel and length can be measured. So we'll see some of the few details how what about the Comet assay. So before that, let's see what is the OECD guideline for this Comet assay. The OECD guideline is OECD 489. Okay, it was adopted in implemented in the year 2016. The title of the essay is In Vivo Mammalian Alkaline Comet Assay. So this particular picture is also taken from OECD guideline 489, which states some of the examples for positive control substances and some of the target tissues. So you can go back, read OECD 489 guideline in detail. In that you will come across this table which shows some of the examples for positive comet substances, control substances, I mean to say. Then next, let's see some of the important what is Comet assay. DNA strand breakage can be measured by alkaline elution and electrophoretic method. As in the first picture you can see electrophoretic method. Okay. Single cell gel electrophoresis also called Comet assay. Okay. Another name for Comet assay is single cell gel electrophoresis. Is a widely used rapid method of measuring DNA damage. Okay, in this essay, in the Comet essay, cells are incorporated into agarose on slides. Okay, lysed, they are broken into small pieces. Cells are lysed so as to liberate their DNA and subjected to electrophoresis. Then that slides are placed under electrophoresis, typically under alkaline condition at a pH greater than 13. That is why it is called single cell gel electrophoresis, to allow detection of the DNA double strand breaks, single strand breaks. So if you see in our first slide, the question contained option DNA double strand and single strand breaks. But here we can also find that it also used for alkali labile sites to identify DNA and DNA protein crosslinks and incomplete excision repair sites. So Comet assay can also be used to identify this kind of damage also. Okay, let's see, the DNA is stained with a fluorescent dye. That's why in the first picture you can see green colored dye for observation and image analysis, right? The as broken DNA fragments in the tail migrate more quickly than larger pieces of the DNA of the head. A blur of fragments. A comet is observed when the DNA is extensively damaged. So that migration you have seen in the first picture. The extent of DNA damage can be estimated from the intensity of the comet tail. Based on the intensity of the comet tail, you can identify relative to the total intensity head plus tail that reflects the amount of DNA breakage. As we said in the first slide, we have to read OECD 418 guideline in detail. Okay. Variations in the procedure permit the preferential detection of double strand breaks. Okay, under neutral conditions. Here it was alkali conditions. Under neutral conditions, double strand breaks or inclusion of digestion of lesion specific DNA repair enzymes in the procedure allows the detection of various DNA base alterations. So what it is saying that from the classic Comet assay method, if you do some kind of changes, you can identify other type of damages also, it states. Okay, such as oxidative base DNA damage. Variations in the procedure permit preferential detection of double strand breaks. So that you have to identify. See, Comet assay, there are modified Comet assays also there, Comet chip assay is also there, that you will see based on the alkali conditions in the neutral pH conditions also, right? So this is the principle of the Comet assay. Cells from an exposed animal or cells from an in vitro exposure, mix cells with warm low melting point agarose and coating frosted microscope slide. This is the slide. Agarose layer with exposed cells um, embedded in a solid agarose. Okay, cell lysis and alkaline unwinding of the DNA takes place. This DNA remnants from the nuclei in agarose slide. This is the slide. Electrophoresis staining and examination. This is the slide reading you will see after staining the slides. Microscopic or imaging. Microscopic assessment by trained harmonized staff. This is very important point. And you can see here Comet data collected by image analysis. Tail movement or olive tail movement, right? Migrated DNA into tail length. Tail length of DNA migration, smallest detectable DNA in micrometer. Percentage of tail DNA, amount of migrating DNA from the nucleus. Okay, this is the tail length, tail DNA percentage, tail movement in micrometer, which is nothing but how you are calculating by tail length into percentage of the DNA in the tail. Okay, so let's see some, as I said, there will be some modification to the Comet assay. So we'll discuss in this particular slide about Comet chip assay. Comet assay has also been modified to detect DNA methylation stat. Apart from single strand and double strand damages, it can be used to detect DNA methylation status. Finally, high throughput Comet chip technique, this is the assay based on traditional Comet assay, simultaneously follows DNA damage load over time in different cell lines in response to various genotoxicants. So I would request you that please go back and read Clayson 9th edition chapter 9 genetic toxicology in page 519 about Comet assay has been clearly discussed. Okay, only one paragraph is there on page 519. Please go and read. Okay, that paragraph only I have kept in this particular presentation. This method qualifies kinetic differential sensitivity and repair capacity within different genetic backgrounds at the level of an individual cell. Okay, although the Comet assay has proven to be a sensitive indicator of DNA damage with broad applicability, it is prone to yield false positive and unpredictable findings under conditions of physiological and mechanical stress. Here again you have to remember is that all the essays, no, they might give some kind of false positive results also. So similarly in the case of Comet essay also, there might be some false positive or uninterruptible findings might be there. That is because of physiological and mechanical stress. As I remember during my master research, I was very clearly warned by my seniors that you have to carry out this, particularly reading of the slide should be taken place in the dark only, because light might also cause DNA damage. So we never know if the DNA damage is because of the chemical or light sensitive. Light sensitive Comet assay is there, so one of the disadvantages you can say. So you have to be very careful. So that shows about physiological stress. Okay, light induced DNA damage or chemical, so we have to be very careful in that case. Okay, then next, it has been used, Comet assay has been used most commonly with human lymphocytes and other mammalian cells, but it can be adapted to diverse species including plants, worms, mollusk, fish and amphibians. So this is very important. Comet assay can be extended other than human lymphocytes and other mammals to lower species, diverse species, plants, worms, mollusk fish, and amphibians, which can be used to do environmental genetic toxicology. As you know that nowadays ecotoxicology is also a very important prime concern, and we have US EPA is the regulatory agency which takes place of ecotoxicology. So extending our Comet assay to identify this kind of ecotoxicology damage is also by including this type of species is also very important. Applicability of the Comet assay and other DNA damage assay to rodent testis makes these method helpful in interpreting risk to germ cells. So in the list we'll discuss now further table 9.2, all the studies, genotoxic studies we will discuss. In that one will be there. One section will be about the damage to the germ cells. In the Comet assay application in the testis identification is also there, that we will see. So this is the Comet chip assay, the high throughput single cell gel electrophoresis assay for measuring the DNA damage. This is the Comet chip where wells are there. Okay, 33,600 on Comets, 350 micropores per well, up to 350 individual cells and well. This is the reading that you can do. Okay, let's see what this about this tail movement is equal to tail length into percentage of the DNA damage. We have already learnt in the previous slide. So what are the characteristics of the Comet test? We will see biomarkers of exposure and or effect. DNA damage is not fixed mutation, so by this assay you can identify DNA damage, but you cannot identify mutation. This is a difference, main important difference. And endpoint is, as I said, it is DNA damage, not mutation. So let's see what are the advantages of Comet assay. Cell approach can be done with Comet assay, applicable on many cell types like kidney, liver, etc. Organ damage can be organ related DNA damage can be identified by this Comet assay. No in vitro cultivation step required. Possible estimation of global repair capacity after in vitro challenging experiments. This method is Comet assay is very economical, cheap, fast, simple, and gives some indications about even apoptosis type of cell death also. So let's see what are the disadvantages of a Comet assay. The detected DNA damage does not correspond to fixed mutations. As we said, it only identified DNA damage. Need of internal reference to avoid experimental variations during electrophoresis. Okay, that is important. You need to establish the method has to be established in a certain lab. Okay, historical data should control data should also be there, and the individuals, the persons who are scoring those slides has to be properly trained, and harmonized scoring should be there, because subjective variation should not be there. Scoring is a problem. Yeah, whether if you see irritation type of scoring or sensitization type of scoring like this, no, it differs from subject to subject. Certain thing might appear red to me, but to another individual it might not appear to be red. Okay, it is subjective variation, we keep saying. Slow individual microscopic scoring, as number of cells to be count will be more. Harmonization among scores interpretation. Cell necrosis and cytotoxic potential confounder. Histopathology required for positive results. So as we already know that with only one single assay we cannot confirm a certain finding. We need to be supported with another type of assay. So even if you find some DNA damage positive findings in Comet assay, in particular kidney or liver cells, it has to be supported with kidney or liver histopathology. That is what it is stating. Okay, DNA damage assay, not mutation. This point is keep on repeating throughout the slide. Okay, so this is very important. In the first slide, whatever the ABT has reference, no, that is about table 9.2, overview of genetic toxicology assays. So you have to go back and read, take lesson textbook genetic toxicology chapter, please read table 9.2. So some of the will try to list the essays here. We have essays, prediction of genotoxicity is there. So interpretation of chemical structure, structural alerts to genotoxicity, in silico predictive models, computational and structural programs, MKS, TopCat, DEREC, quantitative structure activity relationship modeling. So first point though, it is talking about prediction of genotoxicity by using certain softwares. What are those softwares? DEREC, Multicase, QSA. Already some people who are working in the risk assessment type of domain toxicology industry, they might are aware of these softwares. You might be using it in your day-to-day life. because certain chemicals they don't have literature. So for that reason we need to use software to predict, because even FDA or certain regulatory agencies they say that please do not do animal test or in vitro or in cell test. If you can get the same data by using softwares, please go ahead with it, because either if you see Derek or Lasa, Sara, these softwares I am myself I'm also using on my day to day basis at my um, so I find that these data generated by these are validated softwares, even regulator accept this software. So that is the reason it is recommended please use these softwares in order to predict the genotoxicity. Next we have DNA damage and repair assays. Under this, what are the assays we will see? A and B are there. Direct DNA damage of direct detection of DNA damage, and DNA repair, recombination, and genotoxic stress response. So we'll see what we have. So we have direct DNA damage. We have alkaline elution assay for DNA strand breakage in hepatocytes in liver cells. Comet assay, single cell gel electrophoresis for DNA strand breakage. Comet chip, non-mammalian comets in ecotoxicology, assays for chemical edX in DNA. So in our question as well as discussion in the first, second and third slides, we have seen that Comet assay, that is the reason ABT has given the reference, refer to table 9.2 for the right answer. And we have learnt about Comet chip also, right? And application of this non-mammalian Comet assay in ecotoxicology, as I said, no, it is very important. Next, we see DNA repair recombination. Under that, what are the assays which are listed? Differential killing of repair deficient and wild type bacteria, induction of the bacterial SOS system, green screen for GAD D45A gene induction in TK6 human cells, unscheduled DNA synthesis in isolated rat, liver, and rodents in vivo, induction in mitotic recombination. So I would like to say that please don't get panic or get stressed that oh I have to remember all these essays. See, since why I am discussing this table 9.2 in detail, because it is already referenced by ABT. One thing you have to keep in mind, and genotoxicity is important subject definitely for any chemical. In order to screen, you have to do certain genotoxic assay, because there is a thing that if a substance is positive for genotoxic, there is in a long term run it might be causing that is the assumption it is there. Not all genotoxic substance will end up into carcinogenic, because genotoxic substances, if you see, there are genotoxic carcinogen, there are non-genotoxic carcinogen also. So let's not deviate from the topic. The important thing is that can I remember all these essays? I would say it was difficult, because those people who are working in the genetic toxicology domain only, probably they might know all these essays. If you look at a inhalation toxicology or a systemic toxicology risk assessment, he might not be aware of all these essays also. I myself have not come across all this type of essays in my 8 to 9 years of my career span. So let's try read it to 4 to 5 times, because even one question is also in for us important for us to qualify in the exam. So table 9.2, maybe 2 pages is there in the lesson. So we'll try to remember as much as we can. At least some of the important 5 to 6 essays we will remember, so that at the time of exam it will be very easy for us to identify the options and select the right option. Okay, let's see prokaryotic gene mutation essay, bacterial reverse mutation assay, salmonella mammalian microsome assay, Ames test, E. coli tryptophan, WP2 tryptophan reversion assay. So we have various types of Ames assays also given. Then we have assays in non-mammalian eukaryotes, plant assays. Okay, coming to non-mammalian, we have fungal assays and we have plant assays also. Okay, from this you can understand how these are important. Then next, not going to discuss everything, because you list out because I'm already giving the pictures, I'm giving the references, you go back and read. And essays in Drosophila assay. So we have seen fungal assay, then we have now we are seeing Drosophila assay also. Then we have mammalian gene mutation assay. Okay, then in which we are seeing forward mutation assay. Important thing here is also mouse spot test is important. Pick a mutation assay, because when I do literature search I came across this type of test also. Okay, important thing, TK mutation in mouse lymphoma assay or human cells. HPRT or XPRT mutation in Chinese hamster or human cells. This is also very important. Okay, coming to transgenic assay, they have listed out based on LAC-I or LAC-Z gene Okay, coming to transgenic assay, 6 months transgenic mouse assay in case of cancer study, that is very important. Okay, this assays try to remember the genes FAG 174, PKZ1 mice. Try to remember. Okay, no need of going in detail and identify how this assay is performed. What is important is type of assay, type of DNA damage or mutation it is used to detect. Okay, coming to mammalian cytogenetic assays, important thing is chromosome aberration. Read chromosome aberration. There will be specific OECD guideline for chromosome aberration. Definitely we'll try to make another video on specific videos, separate videos on chromosome aberration, micronucleus assay as well as Ames assay. This video we are only listing out which are given in the table 9.2. Okay, micronucleus is also important. Okay, then sister chromatic exchange, aneuploidy in mitotic cells. Okay, then next we have other assays. As I said, the last slide I said on germ cell mutagenesis. Here these type of assays, these listed assays are used. See, Comet assay in sperm and gonadal tissue. One of the slide it had no sperm testis. It is used to identify Comet assay like that. Then you try to, and here also the alkaline elution assay for DNA strand breaks in rodent testis. So these are the, and this is also important FISH. This is fluorescent in situ hybridization test. So try to remember these assays, what type of damage they cause, damage or what type of DNA damage to identify this specific assays. So here pictorial presentation of chromosome aberration as well micronucleus FISH is given. So hope from this video you were able to learn about Comet assay, OECD 489 guideline. Okay, and we have seen advantages and disadvantage. Advanced Comet chip assay we have seen, and we have also very quickly gone through table 9.2, which lists all the type of genotoxic assays and type of damage, DNA damage it to identify those assays. Okay, thank you so much. Be curious. For any questions, please comment and mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel Toxicology on Board. Thank you.